Right, my name's Andy Newhouse, and like came up briefly earlier, the group or the institution I work at is kind of a long name. Has the longest university name in the U.S., I think. It's State University of New York College of Environmental Science and Forestry. So we call it ESF, for Environmental Science and Forestry. And so there I got my master's degree actually working on American elm trees, a similar project with elm. And about the last 10 years I've been working on chestnuts. And I've had the opportunity to work on almost all the different aspects of this project, from working with DNA in the lab to actually maintaining our field plots and arranging some of that. And now I'm actually working mostly on the federal regulatory part of this, and I'll get into that in more detail later. But that's taken the form of a PhD project. So after finishing my master's, I thought I was pretty much done, just like doing the research, and ended up getting back into academia. So anyway, going to be talking about our work with the chestnut, but and also some applications for how that can be applied to Chinquapin. And at home I have twins who are almost turning 5, so that's fun.
You get the award for the best picture today.
They're not in a chestnut tree, unfortunately. Ash, which are rapidly disappearing in our area, so another reason to keep going with projects like this. All right, and given the kind of dubious honor of talking right after lunch, I'm going to give you my 3 main points. So if anyone does need to take a nap, at least you get the important stuff ready to begin in here. So I'm going to start first point here. Chestnuts and Chinquapins are really cool and worth saving. You guys already know that. I'm not going to spend a lot of time on it, but number 2, OXO, as Steve called it, or transgenic chestnuts that have this gene for OXO, can protect American chestnuts from the effect of the blight. And they can do so safely without increased risks to people or the environment. So I'm going to get into that a little bit, some of the studies we're doing. And 3, it seems very likely that this should work for Ozark Chinquapin too. I'll kind of caution you, I guess, or give a caveat that it might take a while, and it might cost a lot of money, and it might be very complicated, more than we think. Or it might be easier than we think, because we've laid a lot of the groundwork with chestnut. Many of the parts should kind of flow into that. So I want to be optimistic, but also realistic about this. So with that said, American chestnuts were cool and historic, and people could eat them, and they have informed songs and street names, and wood was, as you all know, rot resistant and all that, and of course wildlife of a variety of types relied on the trees and the nuts, the consistent mass crop, and you guys know all of that.
Do brook trout eat the nuts, or what's
Good question. The trout there refers to a study that we did with aquatic insects eating chestnut leaves. So a collaborator of ours did this study with several different types of leaves in these little simulated stream environments and bubbling glass jars to see what types of leaves insects would eat. And it turns out in this one study, the insects preferred chestnut leaves to the oaks and the beeches and the other things that have kind of replaced them in the ecosystem. So, right. Trout don't eat chestnuts, but they eat the insects that eat the chestnut leaves, and it's it's part of this keystone species concept that really works its way into the whole ecosystem. So, American chestnuts were huge. Some of these historic pictures, that's a logger there. Some of these 6 and 8 feet diameter trees, really impressive. Another little guy standing here by the very big tall forest tree. This picture is up earlier, a whole family in front of a chestnut tree. Chestnut trees, American chestnuts, could also take a wide spreading shape instead of just the timber type. These are a couple pictures of old chestnuts that grew in open fields. They had this spreading form. These were actually taken in the 1980s, post-blight, and both trees have since died from blight, but really amazing trees. Of course, chestnut blight changed that. And here I'd like to talk just a bit about the mechanism, why chestnut, or how chestnut blight kills trees. And Colton already introduced this. And one of the main ways is that the fungus secretes an acid, a toxin that kills the living cells under the bark. And that's called oxalic acid, or oxalate, and it lowers the pH. It makes this acidic condition that kills the tree tissue. And then the fungus comes through and kind of eats the dead tissue. And what happens on, say, Chinese chestnuts or even oaks, the fungus can survive on oak trees also, but it doesn't do a lot of damage. And it survives there as what's called a saprophyte, where the fungus lives on the tree, kind of hangs out until a branch dies or the tree dies from some other reason, and then the fungus eats the dead wood. But on American chestnut, it's just, and Ozark Chinquapin, it's just very susceptible, and this acid causes a lot of damage and allows the fungus to eat the dead wood sooner, or allows the fungus to kind of actively kill the tree and then eat the dead wood. So fungus growing on the plate there.
Smiley face on that?
Yeah, the fungus is happy to attack the trees. And then here's the sciencey version of oxalic acid. The enzyme that we've added, called OXO, like Steve mentioned, stands for oxalate oxidase, breaks down the toxin, breaks down the oxalic acid, and it forms hydrogen peroxide and carbon dioxide, both of which are used by the tree for other purposes. But that's a little too sciencey, so we made up this comic of Buster Blight shooting oxalic acid at Charlie Chestnut, but we've given Charlie Chestnut an oxalate oxidase shield, and it breaks down the oxalic acid into harmless byproducts. Charlie is safe, Buster Blight is not dead, and they have to learn to live in peace. So, this enzyme that we've put in, oxalate oxidase, came from wheat, and a very important point is that it doesn't kill the fungus. It's not a pesticide. It's not killing this. It's breaking down the toxin that the fungus uses to kill the tree, but it doesn't kill the fungus. So one important point is that that reduces the chance the fungus will develop resistance. So you've probably heard of like antibiotic-resistant bacteria in a hospital or something like that. And in that case, if you're using an antibiotic, you're killing, say, all the weak bacteria. You're killing most of the bacteria. The only bacteria that survive are the strong ones that can kind of tolerate the antibiotic. And that's how you get antibiotic-resistant superbugs, or bacteria. In this case, we're not killing off the fungus. So that selection pressure isn't there. It's less likely that the fungus will overcome this enzyme and develop resistance to it. Another interesting point that was brought up at a meeting I was at earlier this spring was from someone who was representing a Cherokee tribe. I was wondering if Mark might be able to kind of speak to this too. But one concern was that, you know, in the Cherokee view, I guess, that all living things are important, and if we don't think it's important, we just don't understand that thing. You know, if plants have 14 uses, is the thing we heard today. But even the fungus, okay, they're concerned that we might be killing this fungus or killing pests if we don't understand all of what's going on with the pest. Okay? In this case, we're not killing the pest. We're breaking down the toxin that the pest is using to kill the tree. So it could kind of ease some of those concerns also. We have an easy detection method to tell whether our tree, whether a leaf has this enzyme, whether it's transformed. And then this same enzyme, well, we found it in wheat. We used the version from wheat. It's also found in a number of different foods. We have tables as part of our submissions to the regulatory. But strawberries, bananas, cocoa were a few of the common ones. In the wild, several wild grasses related to wheat, but also things like moss. There are a few different fungi that also produce this same enzyme. So the fact that we're putting it into a chestnut, it's new to the chestnut, but it's not new to our diets. It's not new to the environment. We're not introducing something foreign necessarily to either diets or the environment. And Colton talked about one way we could kind of screen for whether this is working, whether, you know, putting the enzyme in, whether it makes a difference, whether it works. I'm going to introduce a little bit of a different way. The real traditional method is to take a bigger tree, at least a few inches in diameter, take a chunk out, like Steve was saying, a silver dollar chunk, and put some of the fungus in that, and then wait all summer. We have kind of a modified method where we're testing trees of maybe a ½ inch diameter or so. We use this little wire hook. It's called a nitpicker for fixing a sweater or something, I guess. But anyway, it's a tiny little small diameter wire hook. You kind of push it into the soft bark of these young trees and drag it down. And that just creates a wound of a consistent depth. And then, you can't really see it on the picture here, there's a little scratch in the bark, and then we take a plug of the fungus, or the agar with the fungus growing on it, put it on that scratch, wrap it around, wait maybe 6 weeks on these small trees, and then we can either measure the canker or see whether the tree has died. So this is just another way to screen whole trees. It's a little bit more a little bit more realistic than the leaf assay. The leaf assay is a good predictor, and this is just kind of more of a sure thing that, to screen trees when that's a possibility. So this is a greenhouse assay we did with 6 trees of each type. On the far left are the non-transgenic American chestnuts. They all wilted. You can see a little bit of new growth down here. That's from below the canker. They're re-sprouting as they do in the wild. The transgenic American chestnuts in the middle are all doing great. They haven't even wilted. And then we have our Chinese chestnut controls on the right, and half of those died in this experiment. Because on these real small stems, it's a very harsh test. When we introduce a relatively large load of the pathogen, of the fungus, it can kill even our resistant controls. But all of our transgenic American chestnuts are doing great. And we still have some of these same trees in our greenhouse producing pollen for other experiments and so on. So the greenhouse experiment was looking really good. And then another field experiment. These are trees that are actually full siblings. So this is from transgenic pollen. We grew the offspring of pollinations. I'm going to talk a bit more about that in a little bit. So these 2 trees came from the same parents. One transgenic parent, one non-transgenic parent. Guess which one inherited the transgene? It's a tiny little, that's probably mostly the result of the wound. But there's a little bit of fungus. There's some orange tissue along the sides there. So the fungus is still growing there, but not killing the tree. Like we said, we're disabling the fungus. We're taking away the weapon the fungus uses to kill the tree. And this is what happens on a typical susceptible American chestnut. Just lots of damage. When we took this picture, the tree was still alive, but it clearly wouldn't have survived the next season. It was almost fertile. A lot of damage from the blight there. Okay, yes, that one has the OXO transgene. Okay, so after we know this works, we see these trees are highly resistant to the blight, we want to be sure they're safe, that they're not going to cause any harm to people or the environment. So this is actually a subset of some of the studies that we've done, and I don't have nearly enough time to go through all of them, but I'd be happy to maybe answer questions later about some of these. But I am going to go through to a little bit of detail on the first one, tadpole growth and survival. I'm going to get back to that. But other studies we've done, mycorrhizal colonization of the roots. We want to make sure we're not interrupting beneficial fungi or relationships there. We're looking at bee feeding on transgenic pollen, caterpillar feeding on transgenic leaves, aquatic insects like I mentioned, decomposition of transgenic leaves. We want to make sure that interactions with other fungi, even decomposing in the forest floor, isn't going to change. Native seed germination through leaf litter was a fun little greenhouse experiment where we crushed up leaves of different types and put those in. What types of plants tend to grow around transgenic trees when we have them planted in test plots? Growth rates and forms of transgenic trees compared to non-transgenic comparisons. Metabolomics is looking at small molecules or enzymes or proteins or various sugars and things that are produced by trees, and we have that analyzed in a number of different tissues. Inheritance patterns from pollinations with transgenic pollen, and getting back to that. And then nutrition of transgenic nuts. And almost all of these, we have, I mentioned here, other species controls for context. We're not just looking at a transgenic American chestnut and a non-transgenic American chestnut. I mean, that's the comparison that we want to make. But in some cases, we would see a difference there. And what does that difference mean? Because often, if you just take 2 random American chestnut trees, you'd see a difference. So we want to make sure we include some other non-transgenic American chestnuts, and then also some hybrid chestnuts and Chinese chestnuts, so we can kind of get some sense of scale for if we see a difference, what does that mean on the landscape for chestnuts that are already out there? And if the difference we're seeing, okay, if our transgenic chestnut's here and non-transgenic is here for whatever measure, but then Chinese chestnut is here, okay, this level of difference doesn't mean anything because Chinese chestnuts are already out there, or whatever the scale, whatever the comparison is, we want to make sure we're looking at the natural variation, or putting the changes in the context of natural variation that's already out there. So, lots of different types of ecological studies going on. Like I said, a little bit more detail about tadpoles.
I have a question for you.
Sure.
You can back up one. That's pretty impressive, y'all doing all this. I mean, that's really good. And I know we don't have enough time to go through each one of them, but Andy, in your opinion, was there any place or any way where you had the pollen, the leaves, or whatever, there was a negative? Was there any negatives found in any of those categories or broad spectrums?
Yeah,
of
that's a good question. And like I said, we're putting all these in context. Any changes that we have seen, we've seen bigger changes in Chinese chestnut or traditionally bred hybrids. So, which are not regulated. So, you know, as the regulators are assessing this, and okay, there's a small change. Well, compared to a hybrid chestnut that anyone can plant wherever they want, that the change is not significant.